Aardvark uses composite visualizations of trees, time series, and images to help researchers understand how cancer cells grow, divide, and spread. Our collaborators collect time series microscopy images where cells are tracked over time. Derived attributes, such as cell mass and cell location, are measured, resulting in time series data. And when cells divide, the relationship is recorded to produce a tree or cell lineage. Each of these data types represent a different aspect of the data, and visualizing them independently cannot tell the whole story. For instance, simply plotting the mass over time may show one cell growing, dividing into 2, then 4. But what if the line chart shows an outlier? In this chart alone, we cannot verify if this is a cell growing abnormally large or an imaging error. To answer this, we need to find the corresponding cell in the images. In another example, we see a divergence in the cell growth. But again, we cannot tell in this view if the faster growing cell divides into the faster growing offspring. To do this, we need to find the corresponding cells in a tree view. Our collaborators are frequently cross-checking between visualization views, but this requires mentally linking separate visual data elements together. Therefore, our work focuses on easing this burden by showing all 3 data types together in a composite visualization. But how do we actually combine these disparate types into a seamless visualization? Let's look at our first composite visualization to explain our design principles. Here we select trees as the primary data type, which we show in a node link representation. This visualization will then host our secondary data types. We can then choose a composition technique to embed the client visualization. In this case, we nest the time series data inside each node. Alternatively, for images, we superimpose them along the nodes automatically and on demand. In Aardvark, we use horizon charts to represent time series data. Image snippets of cells are superimposed above the horizon charts. We place images on the tree topology near division points and where large changes occur in cell attributes. As the size of the tree changes, additional cells are added or removed. Mousing over the horizon chart shows thumbnails with a full cell boundary beyond the clipped region. Hovering over a cell image shows the previous and next frames, and selecting it pins those images. In this case, the images reveal a missed division. The cell snippets show that the cell divided, but one of the daughter cells is tracked. The second composite visualization uses the time series data as a primary data type. A line chart is well suited for comparing the development of multiple cells. The tree data is shown on demand through connecting lines and styling. Images of cells are shown on demand above their corresponding data points. Here, each line represents the mass of a single cell over time. Hovering over a line will show the cell at that point in time. Selecting a cell will display connections explicitly with a dashed line and uses color to differentiate cells in the 2 branches. Here we can see a divergence in the growth between the green and violet branches. The final composite visualization uses images as the primary data type. Showing the images directly helps reveal the spatial relationships between cells. Cell positions over time are superimposed directly in this coordinate space. Cell relationships are shown with a node link representation. Leaf nodes coincide with the cells in the image, and internal nodes represent cell ancestors. In Aardvark, we can see how this looks as one cell divides into 2, then 4, then 8. As the number of cells increases, we can see when they begin to overlap, resulting in issues with tracking. Together, these three views comprise Aardvark, an open-source visualization system for trees, time series, and images. Thanks for watching, and remember, with Aardvark, the answer is just a nose length away, so keep on digging.